Whoops, my bad. Continuing on, is the microphone on? Yeah, it's on. Nucleus contain. Where was I now? Where do proteins of rough ER? Okay, so ER consistent with what is consistent with the second nuclear membrane, or the two types. There's smooth ER, rough ER. The difference between them: rough ER has ribosomes on it. Where do proteins start the rough ER go? What process follows? They go to the Golgi apparatus. Golgi apparatus will modify them, and then they'll just go, protein modification. Function of smooth ER. Smooth ER. What's the function of smooth ER? It's for lipid and steroid hormone synthesis. What's special about it? It's called the sarcoplasmic reticulum in muscle cells, and then it also breaks down toxins and drugs. After synthesis, where do proteins go? Like I said, they go to Golgi apparatus. Protein synthesis occurs there. Protein modification occurs there. Two phases of Golgi and function. The two phases, there's a cis side and trans side. Cis side will cis side is like the receiving side. Trans side is where like the proteins leave in vesicles. Okay, let's go. 3 functions of lysosomes. How do they do it? What made by what? So lysosomes, they're responsible for apoptosis, autophagy, and I pronounced that wrong. Apoptosis, autophagy, I don't know. And they break down nutrients, cell waste, and toxins. And they're made by the Golgi apparatus, I think. Or I don't really know. Maybe that's just something I just... Oh yeah, they are made by the Golgi apparatus. Contain apoptosis, autophagy, nutrients, pathogens, and cell debris. What are they made? Oh, that's what it means by made by what? How do they do it? They have digestive enzymes on the inside. They just they eat it. They have a low pH. Special application of lysosomes. So I'm not too sure about that. Peroxisome functions plus common area. They're found in the liver and kidneys. Peroxisomes, they form H2O2, hydrogen peroxide, and they break down fatty acids and amino acids. Generate hydrogen peroxide to oxidize substrates. Okay, vacuoles, they're just vesicles with the membrane, right? So 4 types of vacuole. There's storage vacuoles, not storage. There's food vacuoles. So they have food and they bind to peroxisomes they bind to lysosomes to digest them. It's called a central vacuole that contains water, like turgid vacuole. Yeah, central vacuole, tonoplast in plant cells. Then there's contractile, which is found in protists, and those release water out of the cell. And there's transport, which moves protein between organelles. 4 functions of the cytoskeleton. Maintain shape. They help move the cell around. They help move the components of the cell around. Some motility, maintain shape. And they're used for adhesion, attaching, anchoring membrane proteins. 3 components of the cytoskeleton, each made of what? So there's microfilaments, microtubules, and then intermediate filaments. So microtubules are made of tubulin. Main function, some motility, moving the cell around. No, that's filaments. Filaments are some motility. They're support and motility, so within the cell. Intermediate filaments, those are used for keeping cell shape. I don't know what they're made of, doesn't say in the notes. Microfilaments are made of actin. They help with cell motility, but I'm freezing, I don't know why. 3 structures in form of microtubules. They make villi, flagella, and centrioles. I don't know. Microtubular arrangement. Okay, basal body is 9x3. The cilia and flagella, the top part, the edge part, those are made by... There's a central part, and then it's 9x2. Yeah, 9x2, and then and then the other one's 9x3. Okay, so it's triplets. Nice. Microvilli composition, they're made of actin. They function to increase surface area. Centrioles are microtubule organizing center structure. 9x3 is similar to the basal body. Paracentriole, centrosome. What are MTOCs? They are they comprise the basal body of cilia and flagella. What else do they do? Form spindle apparatus during cell division. Okay. What organelle makes chemical energy plus two functions? Mitochondria makes chemical energy. The two functions are beta oxidation and like making ATP. Structure of the organelle. There's the outer membrane, inner membrane, the intermembrane space between the outer and the inner, and then there's the intermatrix, which is within the inner membrane. Unique of organelle. It contains their own ribosome. It contains its own DNA, like genetic material. Organelle present in cells, chloroplast function, make ATP, gives the plant color green, absorbs green and red, and then it releases green. Where would this organelle be derived from? Cyanobacteria. Reason for it contains its own DNA. 3 of origin, 3 evidence is strongest. So the 3 points of evidence is that it contains its own circular DNA. The other one, it replicates individually via binary fission, and the third one is contains its own ribosome, I think. The strongest evidence is that it contains a circular DNA. I know that one for sure, but contain other structures similar to bacteria. Okay. Difference of plants and animals. I'm not too sure about that. Organelles are only in plants, so there's plastids like chloroplasts, there's like vacuoles, and then there's cell wall. 2 main classes, membrane proteins, how to remove. 2 main classes, there's integral and peripheral integrals. There's also like integral proteins and then peripheral proteins, and then there's transmembrane proteins which go all the way through. Transmembrane proteins, they're hydrophilic, so they go within, like they just like align themselves with the integral proteins are hydrophobic. They don't like water, so they stay in between the cell membrane and they just stay in the hydrophobic area. How to remove them, use detergent and it'll like just take it out. Then there's peripheral proteins, which just are hydrophilic. They just sit on the outside of the membrane. They're polar and they just chill there with hydrogen bonds. To remove it, you just change the pH concentration, salt concentration. 5 types of membrane proteins. There's adhesion proteins, transport proteins, enzymes. I don't remember. Transport protein, receptor proteins, glycoproteins, enzymes, adhesion, and anchor proteins. What do you need? Two types of transport proteins. There's channel and carrier proteins. Channel proteins, they have ions. Yeah, I don't know. 4 types of channel proteins. Other examples. There's channel proteins, voltage gated, mechanically gated, and ligand gated. I don't remember that one. 3 types of junctions. There's tight junctions, gap junctions, and anchoring junctions, I think. Gap junctions are used for communication between cells. Gap junctions are used for... Wait, I just said that. Gap junctions are used for communication between cells. Tight junctions are used for keeping the things together, which use desmosomes. And then anchoring is hemidesmosomes to anchor towards one thing. Cell orientation, there's apical, that's like facing the outside, like just a vacuum of space. And then there's basal, which is just what side is anchored to it. 2 categories of transport, active and passive. 2 types of passive, simple and facilitated. Degrees of active transport, primary, secondary. Primary is when you use ATP, secondary is when you use the existing chemical gradient. 2 types of cell movements, endocytosis, exocytosis, ways to trigger exocytosis using calcium. 3 types of endocytosis, which uses, so the special protein is called cafrin. Endocytosis, 3 types is pinocytosis, receptor-mediated endocytosis, and Phagocytosis. Phagocytosis is when the cell extends outside using actin filaments to take something in. Pinocytosis is when you just pinch in and take it, and then receptor mediated is when you use clathrin protein binded with receptors to take specific things. Key difference between osmosis and diffusion. Osmosis is when water molecules move. Diffusion is when the other thing moves, like the solute moves. Osmosis is like the solvent, it moves around. Three terms that describe. So there's isotonic, hypertonic, and hypotonic. Hypotonic is when so if you say the solution is hypotonic, that means there's like less concentration of less concentration compared to the other thing. So it's like if you have a red blood cell, you put it into a hypotonic solution. Hypotonic means it has less concentration, so you have less concentration outside, so you want water to go out from the cell, and it will cause the cell to shrivel and not be good. Hypertonic is when there's higher concentration outside, so then the water will go inside and it'll blow up the cell, right? Hypotonic, that's what I said, right? Hypotonic is when there's lower concentration outside, so hypotonic cell. Hypotonic means there's less concentration outside, so there's more concentration on the inside, so the water will want to go into the cell and it'll rise and break. So for animals, they want to be in isotonic because they don't want to explode, and plants, they want to be in hypotonic because they want to have turgid pressure. Okay, cell wall function. Protection and structure. Cell wall found in plants, protists, bacteria, and fungi. Composition of cell wall in each organism. So plants have cellulose, fungi have chitin, bacteria have proteoglycans. Two categories are, so there's gram positive, gram negative. Positive is when it has a thick peptidoglycan layer. Negative is when it has a thin peptidoglycan layer. It also has this outer cell wall, and it has proteoglycans on the outside. Glycocalyx is like a slimy layer on the outside of cells for protection, adhesion, and red blood cell like vessels or something like that. ECM is found in animal cells beyond the glycocalyx and cell membrane. ECM is composed of proteoglycans, collagen, integrins, fibronectin, and lipopolysaccharides. Reason for each one, not too sure. ECM is found, it's adhesion, cell adhesion. It's used for cell adhesion, movement. It's used for cell adhesion, cell adhesion, transmitting chemical and mechanical, and then support. Okay, it's gonna end right here.